Uh, Good morning, everyone. I'm Danyeong Lee from Seoul National University. Our team, BHI, ranked in second place in this dream challenge. Thank you to the challenge organizers for giving us this great opportunity. Let's start with our thought about the key aspects of this challenge. First, in order to predict the gene expression from the promoter sequence, it is important to capture the transcription factor binding motifs present in the sequence and model their interdependencies or interactions. So we tried to design a model architecture suitable for that. Another key aspect was generalization. There were a few discrepancies regarding given training data and test data. While training data were noisy and followed Gaussian distribution, test data were less noisy and followed uniform distribution. So it is important to avoid overfitting on training data and build a robust model that generalizes well on unseen test data. Now in this slide, I'd like to summarize our approaches to tackle those key aspects. First, in order to effectively capture the motifs of various lengths, we used convolutional neural network with multiple kernel sizes. This method is based on our group's previous works, DeepBAM and DeepHigher, which utilized the CNN with multiple kernel sizes to capture the motifs on the protein sequence. Also, we used a sandwich-like architecture consisting of CNN, LSTM, and another CNN. This structure is designed to capture the complex motif syntax patterns step by step. And to make our model generalizes well on unseen test data, we used some data augmentation and test time augmentation techniques. I'll talk about each of these techniques in detail later. Finally, in order to get more robust prediction, we applied post-processing that converts the model's prediction to the quantile value among the training data. From now on, I'd like to talk about each of these components in more detail. First, this is the overview of our model architecture. The input sequence is one-hot encoded. The encoded matrix passes through one-dimensional CNN, bidirectional LSTM, another 1D CNN, and dense layers to obtain the final prediction of gene expression. Let me further elaborate on the role of each component. The first CNN block captures the TF binding motifs in the input sequence. Two different kernel sizes, 9 and 15, were used to effectively capture the motifs of different sizes. We think that each of these concatenated vectors came out from the CNN layer, represent the TF binding motifs. These representations go through the following LSTM layer. This bidirectional LSTM is responsible for modeling the soft interdependencies or interactions between TF binding motifs captured by previous CNN layer. These blue vectors that represent the soft dependencies are fed into another 1D CNN layer. We expected that the second convolutional layer will consolidate the soft dependencies between motifs captured by previous LSTM layer into the hard or base pair resolution dependencies. This final representation goes through the last dense layers. The dense layers output the final prediction of expression value. The entire model was trained to minimize the average Hubert loss of the training samples. So, in summary, our model structure is constructed to gradually capture the higher order interactions between TF binding motifs as the input moves through each layer. Now, I'm going to talk about our data preprocessing procedure. Most samples were 110 base pairs long, but there were longer sequences and shorter sequences. For the longer sequence, we trimmed it from the 3' end. And for the shorter sequence, we padded it with the sequence from the expression vector to give more information. At first, we applied padding only on the 3' end, but when this right padding, left padding, and symmetric padding were randomly chosen at training time, the prediction performance has quite improved. We think that this random padding worked as a kind of data augmentation. And another critical element of our framework is the way we deal with reverse complement sequence. It has been widely accepted that when dealing with DNA sequences, reverse complement equivariant model achieves better prediction performance. Among the several methods to build a reverse complement equivariant model, we used a post-hoc conjoint model proposed in the Those work in 2022. To briefly explain, during the training time, we averaged the losses of the original strand and reverse complement strand to calculate the final loss that is used for backpropagation. This is equivalent to augmenting training data with the reverse complement sequence. And during the test time, the final prediction is obtained by averaging the predictions of the original strand and the reverse complement strand. When we applied this technique, the generalization ability of our model has significantly improved. Moreover, we also used shifting augmentation as a test time augmentation. During the test time, for each sample, we obtained 4 augmented sequences by shifting original sequence 1 base pair and 2 base pairs to left and right. We did the same thing for the reverse complement strand. So to obtain the final prediction, we averaged the predictions for a total of 10 sequences consisting of the original sequence, the reverse complement sequence, and 8 augmented sequences. Finally, I'll talk about our post-processing. Our training data follows the Gaussian distribution, and we expected that the model trained with this data would make the predictions following the Gaussian distribution as well. So, in order to make the prediction of our model as unbiased as possible, the prediction value was converted to the quantile value among the training data. When we applied this transformation, the leaderboard score was slightly improved. So, when we combined all our ideas that I have talked about so far, our team was able to achieve 2nd place in the final ranking. I'd like to thank organizers for releasing valuable data and hosting this great competition. I would also like to thank Google Brain for supporting computational resources and Deep Genomics for supporting the conference fees and prep cost. Lastly, this work couldn't have been done without the support of our advisor, Professor Sun Kim, and all other laboratory members. Thank you for your attention.
Any questions for Team BHI?
Hi, thank you for the talk. I was wondering if you have tested the longer padding for your model instead of just 5 base pair, 2 base pairs, both sides. And also, ProModel is known for their directionality. I'm wondering how much of the performance you gained by adding reverse complement sequences to your model.
Sorry, I didn't hear your first question. Could you repeat your question?
Yeah, the first question is, have you tried the longer padding of the flanking sequences from the vector sequence? Because you said you only used 5 base pairs, right? Or the 2, 3 base pairs. Because typically transcription factor binding sites some of them are longer, some of them are shorter, but it may not be enough to capture the binding events at those sites with those shorter sequences?
Yeah, we tried the longer pairing with the longer sequences, but we did not get that good results. And second question was about the reverse complement sequence.
Yeah. So how much performance you gained by adding the reverse complement sequences?
I cannot remember exactly the real value of performance improvement, but I'm sure that performance has improved significantly when adding the reverse complement.
All right. Thank you.
Thank you.